In this video tutorial, I am going to discuss about molecular chaperones. Now, the main function of these molecular chaperones are the protein folding. They helps in other proteins proper folding. Okay. These molecular chaperones, they are also known as heat shock proteins. Now, why they are known as heat shock protein? The reason is that whenever there is increase in the temperature, what happens that whenever there is increase in the temperature, the rate of synthesis of these molecular chaperones is increased. So, there is an increased rate of synthesis of molecular chaperones. So, because of this observation, they are also called as heat shock protein, and in the short form it is written like this HSP. Now, these molecular chaperones, they are comprises of several unrelated proteins. Okay, it is not a single protein, but it is a group of protein which are structurally not similar, but functionally they are similar in that they assist in the protein folding. So, what are the different examples of molecular chaperone? The first example is the HSP70 and HSP40. This HSP70 and HSP40, both proteins are found in the eukaryotes. Similarly, in the proteins also some molecular chaperones are there. So, in the prokaryotes, structurally and functionally similar to HSP70, there is DnaK protein, and structurally and functionally similar to this HSP40, there is DnaJ protein. Both of these protein, their counterpart is there in the prokaryotes. They require ATP for their proper functioning. So, they are energy dependent, and they mainly brings about proper folding of the protein of newly synthesized variety. Whenever protein is newly synthesizing, so newly synthesized protein. So, whenever protein is newly synthesizing, at that time this HSP70 and HSP40 works on them and brings about proper folding. So, sometimes this process also occurs co-translationally. Okay. Now, in the prokaryote, in addition to DnaK and DnaJ, there is third variety of protein that is known as trigger factor. These trigger factors are found only in the prokaryotes. There is no any counterpart in the eukaryote, and this trigger factor also works on newly synthesized protein. Okay, but the difference is that this trigger factor, they do not require ATP. And now this system and this system both are present in prokaryotes and both carries out same work. So, if we develop a bacteria in which this system is deficient but this system is working, then such bacteria can survive. The reverse is also true. If we knock out trigger factor but DnaK and DnaJ are still there, then such bacteria are also survive. But if you knock out both the system, this DnaK and DnaJ as well as this trigger factor, if both are knocked out from particular bacteria, then such bacteria cannot survive at a temperature greater than 30 degrees Celsius. So, this is the importance of these molecular chaperones, right? Now, let us discuss about the second type of molecular chaperones. Here in eukaryote, it is HSP60 and HSP10. This is found in the eukaryotes. The counterpart is present in the prokaryotes. So, structurally similar to this HSP60, there is GroEL protein in the prokaryotes, and structurally and functionally similar to HSP10, there is a GroES protein in the prokaryotes. So, here whatever function which is carried out by HSP60 and HSP10 in the eukaryotes, the same function is carried out by this GroEL and GroES in the prokaryotes. Now, again, this system also requires ATP. They are energy dependent. Here, this system also brings about protein folding, but this especially works on older protein. So, see what happens. Initially, when protein is newly synthesizing, this system works. So this system either completely folds the protein or it may partially fold the protein. If particular protein is partially folded, then this protein is handed over to this system, this HSP60, HSP10 in case of eukaryotes, and GroEL and GroES in case of prokaryotes. So now on this older protein, this system works and brings about proper folding. So mostly these older proteins, they are either partially folded or sometimes they are misfolded. So this is well handled by this system, right? Now the third system, which is exclusively found in eukaryotes, that is HSP90. This is not found, there is no any counterpart in the prokaryotes. So we can say that it is only found in eukaryotes. Now this is also ATP dependent. This also requires ATP. It is energy dependent. And here, just like this HSP60 and HSP10, this acts on older protein, or you can say in the late stage of protein folding. But here it is observed that this HSP90 does not work on all the older protein, but it has special taste for the protein. So HSP90 brings about protein folding only those protein which are involved in signaling. There are two examples of the protein which are involved in signaling and they are properly folded by HSP90. The first example is steroid hormone receptor. And the second example is receptor tyrosine kinase. Now this HSP90, it folds protein in some peculiarity. See, this HSP90, it induces conformational changes in protein. It induces conformational changes in protein. So as a part of protein folding, it induces conformational changes, right? And because of these conformational changes, particular protein, it may either get activated, so it undergo activation or stabilization. So see what happens. If HSP90 works on particular protein, it may lead to activation, and if it does not work, it lead to deactivation. So by this activation and deactivation, it is thought that this HSP90 must have some kind of regulatory role in our body. So it is thought to have a regulatory role in our system. And the fourth system of molecular chaperone is the nucleoplasmins. Now this nucleoplasmins, just like HSP90, they are only found in eukaryotes. There is no any counterpart in the prokaryote. And this nucleoplasmin, it has very specialized function that it is required for the proper in vivo assembly of nucleosome. So nucleoplasmin, it is required for proper in vivo assembly of nucleosomes. And what is nucleosomes? Nucleosomes, it is DNA plus histone. Inside the chromosome, if this is histone protein, then DNA is wound around like this. Okay, so such type of assembly is brought out by the nucleoplasmin. Now see, we had discussed all these four systems, right? All these four systems are a part of molecular chaperone, but this second system is also known as chaperonin. It is also known as chaperonin. Chaperonin is the sub part of the chaperones, right? So all the chaperonins are always chaperones, but all the chaperones are not chaperonins. So this was all about molecular chaperones. If you have any query or confusion, please write it down in the comment section below. Thank you.